Johan. He's going to talk about lipid fingerprints in chronic viral infections, learning from simulation of the viroporin protein of hepatitis C virus. So you can share your screen when you're ready.
Okay, so hello everybody. Again, good morning or good afternoon. It is a pleasure for me to share with you this time some of the work that we do in the lab. Thank you so much to the organizers for the opportunity. I am originally from Bolivia. I am currently an assistant professor at junior faculty at the University of Buffalo. So you can see the beautiful landscape where I'm currently doing all these simulations. In a nutshell, what we do in my research lab is to study the role of lipids in mechanisms of disease. And to do that, we use computational models. Today, I will specifically talk about protein-lipid interactions during viral infections and how these very local interactions end up shaping or changing the membrane landscape in terms of lipid composition in different locations in the cell. This will depend on the virus as well as the place where it is assembling itself. So the virus of focus today is hepatitis C. Our studies are not so much focused on the dynamics of the virus itself, but I'm going to briefly introduce the life cycle of this virus, and hopefully you will see the motivation for the work that I will share with you this afternoon. So this virus affects specifically cells in the liver. It infects the cell, and then it assembles in the endoplasmic reticulum. Hopefully you can see my arrow moving a little bit here at the bottom left of the screen. This protein assembles, and when a patient has chronic infection, it can lead to complications such as liver steatosis, which is mismanagement of fat in the liver, which is the beginning of one of the possible beginnings for non-alcoholic fatty liver disease or NAFLDs. The hallmark of these diseases is fat mismanagement or lipid mismanagement. One of the ways that the cell has to store fat is in lipid droplets, which are these orange yellow circles at the bottom of the screen. Hepatitis C assembles very close to where these fat storages are being built inside the cell. And when the cell cannot use them properly, so store the fat and then use it afterwards when needed, these fat storages start to accumulate, as you can see in the far right of my screen. When that happens, many different signaling cascades start to evolve, and the disease can evolve to fibrosis, sometimes into cirrhosis, which is liver scarring, and sometimes it can evolve into liver cancer. Our motivation for the work I'm sharing with you this morning is because one of the proteins that is important for the virus life cycle, called P7, has this sort of conical shape. Is a channel that forms inside the membrane cells, inside the cell membranes, very close to where lipid droplets are forming. And it has been identified through experiment as a potential drug target to block the replication of the virus, but also to block whatever else is happening that triggers the mismanagement of these lipid droplets in the cell. What we're trying to do, and you can see a picture of the student doing this work in the top right, is understand how this protein, first of all, is interacting with the membrane, how it inserts in the membrane, and then how it oligomerizes to form the channel that is known to have as the final conformation. As this channel is forming, what you can imagine is the lipids and other components in the cell membrane shifting and moving around. And what we want to characterize with simulation is what are those changes in the very local environment of the membrane around this protein, again, when the protein binds, inserts, and then oligomerizes. And what could be these changes in the long term that affect the dynamics of lipid droplets and fat storage and management in the cell. What we're characterizing through simulations is the work that I will share with you today, just a single monomer out of the 6 that the channel has, and how it binds to the membrane. With simulations, we have the luxury of tracking down the positions of all the atoms, and then we can compute physical properties, mechanical properties of the membrane at different radius with respect to where the protein is binding. Very briefly, cell membranes are made up of many, many different lipid types, as well as proteins. Lipids are by far what forms the scaffold of these membranes, and mammalian cells have thousands of different lipid species. On the screen, you can see some of the lipid head groups that are mostly common in mammalian cells. We have phosphatidylcholine, or PC, PE, which are neutral lipids. We also have a small head group that is charged called phosphatidic acid, or PA, PS, which is also a charged lipid, and a very interesting type of lipids that have rings on the heads of the molecule that are phosphatidylinositol, or PI lipids. Also very important for mammalian cells are sterols. Cholesterol is one of the typical sterol molecules in our cells. And different ratios and distributions of these lipid species are what give specific cell membranes different properties that allows them to host different different proteins, which in turn will perform the different functions that the cell needs to remain alive and to stay healthy. With molecular dynamic simulations, what we want to do is model the behavior, the movement of atoms with respect to one another based on the physical forces that act on them. Molecular dynamics is a technique based on statistical mechanics, which essentially tries to understand macroscopic properties based on the microscopic interactions of this, in this case, atomistic level detail. What you see at the bottom are very weird looking equations. The first one you may be a bit more familiar, that's Newton's law of motion. And the second one is a huge long equation that we call the simulation force field, which has the parameters to model the different interactions between atoms. They can be non-bonded, they can be electrostatic, they can be bonded, they can be angles, the vibration between these bonds and angles. And what we can do when we solve this equation numerically, when we run molecular dynamic simulations, is essentially generate a movie. So we have our coordinates, we get coordinates for proteins from the protein data bank through X-ray crystallography or cryo-EM studies. And once we have those physical positions, the XYZ coordinates, we start a simulation. The program will solve this huge equation numerically and essentially generate a movie of how these atoms are moving, given that they are in the vicinity of one another. For those of you who are familiar with molecular dynamics, the systems that I will be showing in the next few slides will run at MPT dynamics, which means constant number of molecules, constant pressure and constant temperature. We run at periodic boundary conditions, which simply means that while we are simulating a small box, simulation box, in which we have a membrane at the center, like you can see on the slide, periodic boundary conditions essentially replicates this in all directions. So it becomes as if we were simulating an infinitely long membrane slab. We run two different membrane models, which we call the control model and the charge model, with different ratio of relevant lipid species for modeling the endoplasmic reticulum, which is the place where this protein from hepatitis C is known to bind, insert, and oligomerize to form the channel. We are running this with two membrane models that both have charged lipids, which essentially means that we have a charged membrane surface, but they have different ratio of charged lipids. And the key difference between them is that the control model has this very weird looking lipid head group that has a ring on it, the PI lipid, and the charge model has the PI lipid and PS lipid, which is simply a different structure. It's a charged molecule, but it doesn't have a ring on the head. We selected these two charged lipids as models for charged lipids in the endoplasmic reticulum based on experimental studies, and very few works that looked at the P7 channel from hepatitis C that also identified these PS lipids, the charged lipids, as very important for the oligomerization of this protein. To make sure that we're not biasing or forcing our simulation to have the protein bind the membrane in a specific conformation, what we did was start our simulations with the protein vertically oriented, horizontally, or vertically but flipped on its axis. So we are starting, you can think of this as separate individual experiments. And the whole point of doing that is that when we do the analysis, we can corroborate that the behavior between protein and lipid interactions is reproducible. So as a very first analysis, we looked at the initial binding of this protein. And we saw that in all of our replicas, we ran multiple times the simulation, again, to ensure reproducibility. We can see the protein binds the membrane within the first 800 nanoseconds. We ran very long atomistic simulations, two microseconds, which is considerably long for this type of systems. And we see that the protein can bind the membrane, both membrane models. It can bind whether it was initially horizontally or vertically with respect to the membrane, but it binds in different conformations. And what you can see right away from the pictures on the slide is that in the control system where we had a lower membrane surface charge, the protein doesn't really go far below the region of lipid head groups, which is represented by these gold spheres in the picture. Whereas the membrane that had more charge on the membrane surface and specifically had phosphatidylserine or PS lipids in the model, the protein actually binds a bit more flat, but it actually goes below the lipid head group region in the membrane. The bar plot that you see on the left of the slide is showing you the hydrogen bonds that are internal to the protein. So we're not looking yet at protein-lipid interactions in this analysis, just at the internal interactions in the protein. What are the interactions that are stabilizing these different, let's say like a ball conformation or more of a flat sheet conformation that the protein has? We see that the number of hydrogen bonds between the control and the charge models shown in blue and orange in the bar plot doesn't have really statistical significant difference because the error bars pretty much overlap with each other. But the average trend is showing that when the protein is bound and interacting with the charge membrane, the internal interactions in the protein between the helices that make up the protein seems to have more number of hydrogen bonds. If you look at the bottom at our root mean square deviation, which is looking at how the protein conformation changes from the very beginning of the simulation to the end of the simulation, you can see that our different replicas stabilize at different points in time, but more or less for the control system, after 500 nanoseconds, the protein is bound and the conformation doesn't change much. We have slightly different bound conformations, but it's very typical to see this sort of ball shape that is hovering, bound to the membrane, but just on the surface. Whereas for the changes in conformation of the protein with the charge model, the one that has PI and PS lipids, there are much more marked differences between the replicas. In all cases, the first amino acids to interact with the bilayer were lysine 1 and lysine 3. Lysine, these are both in the N-terminus of the protein, and lysine is a charged amino acid, which presumably would be interacting with the charged lipids. Something interesting that we decided to look in a bit more detail was this black curve in one of our simulations for the protein with the charged membrane model. And we see that upon initial contact, you can see here shown in the slide the lysine residue, which is interacting with some of the charged lipids in the membrane. And as the simulation progresses, and after the 1 microsecond mark, what is happening is the protein is starting to insert. We only saw this very deep insertion in one of our simulations, but it's very encouraging because what we want to be able to reproduce in the end is the mechanism of insertion of this protein and eventually oligomerization to form the P7 channel to further characterize how the membrane local environment is changing as all these processes are taking place, right? Binding, protein insertion, protein oligomerization. We then examine at how each individual amino acid is fluctuating, is moving as the protein binds the membrane. And what you can see in this slide is that essentially the trend is the same, whether the protein binds a control model or a membrane model that has more charge in the surface. There are very few regions where you can maybe see that the error bars between the blue and red curves are not overlapping, and those correspond to this loop nearing the C terminus of the protein. We looked at the protein lipid interactions, the number of hydrogen bonds between protein and specific lipid species, and you can see that for different lipid species, in this time, especially for PI lipids and PE lipids, we can see a difference between membrane models. And we see that with the control membrane model, where the protein is pretty much shrinked into a ball on the surface of the membrane, there are more contacts, there are few contacts with PE and PI lipids, whereas DOPC has a more number of contacts with the protein. On the other hand, when we look at the protein interacting with a membrane that has more charge on the surface, there are more hydrogen bonds, there are more contacts between the protein and PE, PI lipids, and of course POPS, which is our additional lipid in that particular membrane model. Once again, you see on the slide in the pictures that the obvious difference between these membrane models is the degree of insertion of the protein once it binds to the membrane. We then decided to look at how is the local lipid environment changing as the simulation progresses and after the protein has been bound stably on the membrane surface. So the analysis for protein lipid contacts was done in the last microsecond of simulation, the second half of our simulation, where we know the protein has found its stable conformation. The binding conformation is not changing anymore. If it was a ball when it bound and it shifted a little bit, it remains like that for the last microsecond of simulation. So we are looking now at how either a ball conformation on top of the membrane or a flat sheet on top of the membrane that starts to lower in height, which was the case with the charge membrane. How is that changing the local lipid landscape? What is the local lipid fingerprint when this protein is bound for long times on the membrane? Just as a reminder, our control model had the PI lipid, this bulky, funny-looking ring on the head group, and the charge model had the ring lipid as well as another charge lipid. So it is more negatively charged on the surface. When we look at the number of contacts for the last microsecond of simulation, the protein is already bound, and now we can start to see the lateral movement of lipids in the membrane. We find that the control model seems to be a bit more fluid in the sense that it allows for one of the lipid species to be recruited to where the protein is bound. In this case, it turned out to be the PE lipid. And the charge membrane actually maintains it. It seems to be a bit more rigid. The lipids are not really moving. They are not being further recruited to where the protein is bound. There seems to be a depletion of PS lipids, which was surprising to us. We thought the PS lipids were going to be actually recruited to where the protein was bound. But as the one lipid species is removed from underneath the protein binding site, another lipid species, the red curve in the bottom plot, which is DOPC, starts to be recruited near the protein. We wanted to see how this affects the mechanical properties of the membrane. That's just some images of the final bound conformation of the proteins. At the beginning, I told you that we're interested in ultimately analyzing how the interaction of this protein starts to change the structural and mechanical membrane proteins membrane properties, very close to where the protein is bound versus a mid distance and then far away from the protein. The reason why we want to understand this is because we hypothesize that as this protein binds, inserts, and oligomerizes, the mechanical properties of the membrane are changing, and because and the lipid distribution of the membrane as well. And because this is very close to the sites where fat storages or lipid droplets form in the membrane, it may affect the content, the composition of these lipid droplets, which may determine whether they are big or small, whether they can be digested afterwards or not. And it could be one of the potential molecular mechanisms for why fat mismanagement occurs when you have chronic viral infections. What you see, the plot you see on the slide is a traditional plot for membrane order parameters or deuterium order parameters. We call them SCDs. These are a value between 0 and 0.54. It simply means the higher number you get, the membrane is said to have a more ordered environment, or the lipid tails look like the ones you see in the picture. The lower value you have, it means it's a more disordered environment, or the atoms have more degrees of freedom. How many minutes?
Viviana? Yes, you are almost over the time. So
okay,
if you can summarize up, that would be great.
This is the last slide. This is the last slide.
That's fabulous.
So you can see two curves very, very close to each other, which are difficult to distinguish in the slide. What we did was look at the properties in the red region in the image and then the blue region in the image. So very close to the protein and far away from where the protein is bound. And in the cases with each membrane model, what we calculated was the difference between these two curves, right? That is very, very difficult to see in this plot. What we see was not so much of a difference in PI and PS lipids, which are the charged lipids and were the ones we expected to show the greatest difference. But there is a significantly different trend in PE lipids. And this is actually where we are currently in the research. We're trying to understand why this is happening. In one of the membrane models, we see the lipids, the PE lipids are very nicely and ordered parallel to each other, whereas in the other membrane, they are more splayed, they have more kinks. And this is what we're trying to currently understand. And again, the overall goal of our work is to understand how these protein-lipid interactions are changing the local lipid composition and properties of the membrane, such that when the channel is formed, the local membrane, in this case of the endoplasmic reticulum, turns out to be very different in this sick cell or in this infected cell versus the healthy cell. Right, so once again, I want to thank the organizers and just leave you with this concluding bullet points for our work. Thank you so much for your support and for your attention, and I hope everyone continues to enjoy the conference.
Thank you very, very much, Viviana. Very interesting talk. Is there any questions? Remember that you can put your questions in the Q&A box. Let's wait a bit. Okay, so I just had a curiosity. Sorry, I just missed a part of the beginning of your talk because I was not able to join on time. What is going through the pore of this protein, of this ion, not ion, of this channel? What is going through it?
It is an ion channel for the virus.
Okay.
So it is known when it's in a formed viral particle, or as the viral particle is assembling, it's supposed to be an ion channel. But this is called a viroporin. It's a pore of some sort in the membrane. I don't think it's really clear what it's doing in the membrane when it's not in the virus. And in the viral particle membrane as an ion channel, outside of it, I don't know.
Cool. So isn't there any chance that also the complex is being assembled before being inserted in the membrane?
I don't know if anyone has looked at that.
Okay.
I don't know if
because I know sometimes
that
happen, that you assemble before and then you insert the whole thing.
There is a bunch of studies we want to try. Is if you have multiple monomers near the surface, do they assemble before, exactly your question, or do they have to really be inside to be in contact with each other?
And come together. Yeah, cool. Let me see, I think. Oops. Okay, one question and the last one, and we go to the next one. The question is, has been determined if ion concentration could manage a change in the assemble, such as the calcium? I mean, I guess.
Yeah, I'm certain of that. I think both whatever is going through the channel as well as the lipids around it will modulate that activity. From previous studies in the E channel, which is the viral pouring for SARS-CoV-2, we did see in some simulations amino acids in the inside of the channel flipping outside or flipping conformation inside the channel based on the lipid composition around the channel. So yeah, every little detail matters and affects the dynamics of all the components, right? I think it's just so complex that you cannot pinpoint one single thing at a time. It's very well coordinated.
Yeah, cool. Great. Thank you very much, Viviana, for your presentation.
My pleasure. And congratulations on the programming. It's a very nice conference. Thank you so much.
Yeah. So it is my pleasure to introduce you to our next speaker, which is Ana Julia Vélez Urreda from the National University of Quilmes in Argentina. And she's going to be talking today about CaviDB, a new database of protein cavities that features for structure-based drug design. Ana, over to you.
Okay, thank you, Marcia. Okay, I will try to share my screen. Now I'm being in the opposite side. So it's kind of difficult. Okay, let me know if you can see my presentation.
Present. Yeah, perfect.
Okay, so hello everyone, and thank you for being here. Thank you, Mar, for the presentation. As you may know or not, my name is Ana Julia Vélez Rueda, and as Marcia said, I'm a CONICET founding postdoc at the National University of Quilmes in Buenos Aires. My academic work is focused in the structure and function relation of proteins. And yeah, today I'm going to present you all CaviDB, a database of cavities features in the structural and conformational space of proteins. As you may know, proteins are the functional, structural, and evolutionary units of cells. And we know since Amundsen, I can't see you, so for me it's so tricky. So while I'm presenting, I can't see your faces. So we know since Amundsen's experiment that there is a very strong relationship between structure and function. Nowadays, we know that there are multiple foldings that can explain the functionality of a protein, as Anna said earlier. On their surface, proteins are shaped into many different cavities and protrusions that describe microenvironments that sustain catalysis or ligand binding or protein-protein interactions. And as we said before, since the proteins are moving, the cavities topology is dynamic too and moves too. And this dynamic is central for understanding the protein function. Functional cavities are generally located within the protein domains, and the biological activity of those cavities may or may not correlate with the domain function. And the conservation of these cavities may also exceed the particular domain family. So the knowledge about one particular domain activity is maybe not enough for describing the full functionality of the protein. Maybe we need to have an integrative approach, considering the whole cavities and the whole domains from the protein to study the protein function. So here we present CaviDB, which is a free and open database and tool that integrates very commonly used software for predicting protein cavities and protein features, structural and sequential features. and that can be accessed by in CavityDB.org. And so let me show you how it works in a very short live demo. I hope everything goes well. So here you have, I don't know if you can see the screen. Now I changed.
Yes,
Perfect.
Okay, cool. So here you have the CavityDB UI. The first step for running CavityDB is providing it a valid PDB ID entry or UniProt entry or AlphaFold entry. Here we have the complete PDB database analyzed, and also we have all the AlphaFold models for SwissProt, and so you can access to all that information here. And so once you have entered your PDB ID, or just by picking one of those examples that you have in the home of our database, then you can check or you will have the general information related with your search that consists in the UniProt ID related to the chains that you are looking for, the amount of cavities that were predicted for this chain, and the number of cavities that are druggable. You can access to the complete information by clicking here in the eye. You will have the entire information about this entry split it in 3 sections. The first section is a general one in which you can check all the cross references for the entry. So here you have the entry ID, which consists in the PDB or AlphaFold conformer ID, and the chain ID and the model ID. It's like all jointly, and so this is important for you to search in CavityDB. And so you have here the cross references, and also you can check the structure, the protein structure here in the visualization. Then you will have a second big section in which you have all the information related with the cavities predicted for this chain. You can click here, for example, and then you will have the information related with the druggable predicted cavities. So this criteria that we are using for selecting the cavities is as druggable. Sorry, it is related with the software that we are using, FBucket, which allows you to know if it is a druggable or probably druggable cavity or not. Once you click here, the cavities are highlighted here in the structure and also in the sequence. You can check the information or the specific residues in which the cavity is by clicking here, and you can access to the whole or the complete sequence of the protein. Also check other cavities and in which positions are they. Let me show you here in the bottom. You can access also to the specific features related with those cavities that you are clicking or highlighting. And those features are sequential and structural features coming from different softwares and feature softwares. And you can also download this information by clicking here. When scrolling down, then you can access also to the information related with the interactions or the internal interactions of the proteins and the interactions between cavities. Here you have a plot in which you can check all the interactions and all the cavities interacting. And the highlighted dots are those who were described as druggable or described as druggable. And here you can have, or you can access to the information about the energy, the binding energy between those residues interacting between each cavities. So you can click and search all the information related with all the different types of bounding between, or the different interactions between cavities. And also, as we know, and it is very important to describe those cavities that may act as active sites, in enzymes, that we know that for those specific catalytic microenvironment, shifted residues are important. And so we detected also for all the cavities, which are the shifted residues in each cavity, and you can access to the information here in these plots, in these box plots. So you can compare how, or to check if there is any activated cavity in the chain that you are looking for. Finally, you have a third section here in this entry, in all the entries in CavityB that are related with the global descriptors of the protein, or the solvent accessibility, the complexity, and all the parameters that can be that are related with the global protein information. Going back to the search, we can also we said at the beginning that CavityB also provides information related with the conformational diversity, and allows you to explore the conformational diversity on the dynamics of cavities. So when you are looking for example, for a specific UniProt ID that have more than a conformer in PDB database or AlphaFold, then you can click or select more than one conformer to compare. And so you can compare them and check which cavities are overlapped or not, and if they have also the same cavities, or if they have druggable or not cavities in those proteins or in those conformers. And also you can compare if those cavities that each conformer has are or not activated, and accessing to the specific information for those entries. Also, as you can see, when you have entries coming for AlphaFold, and as you may know, then you can have some regions that are missing in the PDB's conformers that maybe you here in CavityB you can access the information, and you will have also the descriptors for those missing regions in the other conformers. So going back to my presentation, something that I missed is that you can browse our website, and you can check and have all the information related with the search and all the tutorials for that may help you to make different type of search in CavityB, and all the information related with the descriptors that you have for each entry, from where they are coming. And so here you have all the information, all the softwares that we are using.
Anna,
so
just one thing.
Yeah.
3 minutes.
Tell me.
Sorry.
3 minutes.
We have 3 minutes. Yes.
Okay. Then, yeah, I'm closing.
It's a lot of information. Yeah.
Yeah. Sorry. No, just to close and to summarize a little bit what we have in CavityB. As you can see, we have a lot of information here. We have, as I told you, the complete PDV entries, and also we have all the models that were predicted for Swiss protein AlphaFold database. We were able to characterize 36 million cavities in those conformations and those proteins. We have, as you can see, 100,000 almost different proteins in cavity B, so you can access to all this information and check what you need. We really aim with this tool providing you a tool for exploring the conformational diversity of cavities, and also for developing some approaches in the drug discovery considering the conformational diversity of the proteins. If you want to know a little bit more about the methodology, you can read our preprint. We are still in revision for the final paper, and then here you have the paper too. Just to close the talk, I would like to thank the founders of this project, that is CONICET and WBDS. Sorry, it's the I'm so used to use the letters, our letters. So Amazon also founded this project. I would like to mention Toyoko, which is a company that also helped us with the implementation in Amazon too. I would like to thank Franco for developing with me this amazing tool, and to my advisors, Nicolas Palopoli and Gustavo Parisi, and all this amazing group that you can see here that corresponds to my group, my research group in Quilmes, and also to Lily, who you may know, and she is supporting me all the time with checking all the UIs and everything. So thank you very much, all of you, for being here and for listening. Well, I'm open to questions now.
Thank you very much, Anna. An amazing presentation. Really, really good work. A huge amount of work. So we have some questions. The first one is not a question, it's something that I agree with. It's congratulations on the design and the colors that you've chosen. It's really nice.
Oh, thank you.
It's a really nice display. Yes.
Thank you.
We have two questions. Another one also says congratulations. It allows to take useful information together. Which softwares did you use to make that data? I guess they are asking about which programs you use to calculate in the cavities.
Yeah, as you may know, the prediction of protein cavities may be tricky, and there are a lot of tools for predicting cavities, pockets, and whatever. So choosing the tool is kind of tricky, and you will be never enough or comfortable enough with one software, you know. So our intention, or our future work, we are actually working on that, is to retrieve other predictions from other software. By now we are using FPocket, which also provides a lot of features that are very useful. And I didn't told this, but this is a tool implemented in Python, so it's completely implemented in Python. That's why maybe we started with FPocket, which has a library in Python, which is open, so we can use it. And for the features, we used a lot of different libraries and softwares for predicting the contacts and the shifted resids. We used Probka. For predicting some of the structural and sequential features, we use CIDR and some other libraries that you can check in the website, because there are a lot of features, more than I think that more than 30 features, different features for each cavity and for the global protein. So you can access to all the information in our website.
Great. And briefly, two more questions. How did you calculate the contacts? And if it is possible to download the complete report for, I guess, a protein? Yeah.
Okay, yeah, as I mentioned, the contacts were calculated using Probka, which is a software that also calculates the PKI. pKa, thank you, Mark, of the residues in proteins, and so from there we can retrieve also the information related with the contacts, the protein interactions. Yeah, you can download the information, but by each section. For sure, maybe in the future we can and we will implement some new features for the UI, and so you can access also to that information. We are also working to validate these cavities with other databases well known for example, active sites, as you may know, Mark. The database that Shannon Thornton has, and some other databases that are describing binding sites too. So soon will be changed, but it will be improved too. So
cool. Thank you very much. I think there is one more question, but I'm going to go to the next speaker. So if you can answer that one, sure. That would be great.
Thank you.
Yeah.
Thank you very much.
Thank you, Anna. Our next speaker is Sana Bogugerua. Sorry, I'm sorry if I pronounce it wrong. Yes, from the University of Paris-Saclay, and she's from Algeria. Wow, we have a lot of diverse representation today in this Congress. And she's going to be present graph theory for molecular dynamics simulations. So, Shana, up to you.
Okay. So, hi, everyone. It's a pleasure to be among you today. So thanks to the organizer to give me this opportunity to present my research work. So I'm Sana Bogugerua, research engineer at LAMB laboratory at University of Paris-Saclay from France. So today I will try to present the different application of graph theory in molecular dynamics. So it will be like as a post-processing tools for molecular dynamics simulation results, starting by presenting to you the 2D molecular graphs, and then the graph for hydrogen bound rings, and finally another application where we are more in spectroscopy analysis. So the first application of graph theory is the conformational search. So here what we want to do is starting from molecular dynamics trajectory. As you know, in molecular dynamics, we can explore one or more trajectories depending on the temperature and the parameter that we put in the system and the duration of the simulation. And what we want is to have something that give us or that provide us the conformational change in automatic way, because for now what they are doing is to try to visualize the different trajectories and try to see this by eyes or develop like dependent system codes in order to get those conformation. So what I have this work has been initiated in my during my PhD works, and now we are trying to improve this work. The idea is to define the conformation as a mixed graph. So where the vertices represent the atoms of the systems, as I show here in the middle. So we use a colored graph where the color represents the chemical type of the atoms. So here, for example, the blue for the nitrogen, gray for the carbon atom, and the red for oxygen atom. And the edges between those vertices represent the different bonds or interaction. We have designed this algorithm in a way that we can extend to any types of bound or interaction. So we distinguish between the different types. That's why I have here several sets. So I have, for example, set of covalent bound, a set of hydrogen bound, electrostatic interaction. And now we have started our work with a group in the Netherlands where we have added a new type of interaction that we have called organometallic interaction for catalysis. Why it is a mixed graph is because we use directed edges and undirected edges. As you see here, I'm not representing the hydrogen atoms, is to simplify the graph. But for hydrogen bound, we know that we have a hydrogen atom that is involved in this kind of bonds. So to keep the information about which atom is covalently bounded to this hydrogen atom, we use a directed agent, like here. So this directed from the nitrogen to the oxygen. This means that the hydrogen atom is directly covalently bounded to the nitrogen. And the idea here for to use the 2D graphs is that we consider a conformational change if we have a change in the covalent, in the set of bonds or interaction. So here I have put two examples of 3D structures where we have exactly the same set of bonds, but we can see that the atoms have not the same position. From a 2D graph point of view, we'll have the same mixed graph, and then for our algorithm, it will be considered as the same conformer. So a change in conformer for us is a change of bound. So in order to define a conformational change, we use the isomorphism algorithm. In the literature, so we have many methods. We have used McKay, which allows to have a canonical graph, which are signature of those 2D graphs. And then with those, we compare if we have isomorphic or not isomorphic conformation. So as an input of our algorithm, we will have the Cartesian coordinate along all the trajectories, so the different that we call snapshots. So we will have set of snapshots. And for each snapshot, we will construct the 2D graph based on distances and angles to define the different type of bounds. And then we apply the isomorphism test. So here, for example, we see that this conformer is isomorphic to the conformation A. So we do not think, we just add this as a, we increment the frequency of appearance of this conformation. And for example, in the next one, it is not isomorphic, so we will add it to the set of conformation already identified. At the end of the algorithm, we will have the set of conformers with their the different period of appearance and disappearance, and we will get also the different transition between those conformations. So I will show you a small example just to have an idea about the outputs of this algorithm on a small peptide where we have only hydrogen bonds dynamics. So here I have taken a peptide with 24 atoms on trajectory that of 4 picoseconds. And here we have identified 4 conformers. So we can see that the change between, for example, conformer 1 and conformer 2 is the hydrogen bond that disappears. And then we have another one that appears. And at the end, we have a proton transfer. We have the algorithm provides also a global view of the dynamics. And here we have 2 rows. So at left, I have the time evolution of conformation. So we can see through this graph how the conformer R or how the molecular structure is behaves. So here, for example, we can see that the conformer 1 appears just at the beginning, and we have the conformer 2 and 3 that are dominant in this trajectory. At the other hand, we have the graph of transition, which is another view of the dynamics of the trajectory, but here we forget the time. And here I have a graph where the vertices represent the different conformer that I have identified in this trajectory. And the edges represent the transition that have been seen during this trajectory. We have labeled the edges, so we have the chemical type of change. So for example, here I know that we have a hydrogen bond that disappears, and here when we come back is this hydrogen bond that appears. We have also the frequency of appearance, the frequency of the transition. And from this, we can see that we have huge transition between conformer 2 and 3. And we can also have the percentage of appearance for each conformer. And with this information, we can see that conformer 3 is the most present for this trajectory. So we started to apply our algorithm. So during my PhD, we have focused on the peptide in a gas phase, so small peptide and big ones. Then we try to apply it to saccharide for spectroscopy. We also applied our algorithm to molecules in for CID, where here we are interested on the fragmentation. So we try to analyze the different fragments that appears along the trajectories. But the idea is to have something general that we can apply for any system. So we have also applied our algorithm to more complex systems, so especially the condensed matter. And here we have tested the liquid-liquid interface, liquid-solid interface, and we have got interesting results, especially for the organization of water molecules in those interfaces. So I will not show now because of time, but you can go to the papers and see the different results. And now we are working with a group in the Netherlands where we adapt our algorithm by adding an organometallic interaction to try to identify an important event for catalysis. And we have a paper that has been just submitted on that. But the problem that we get with this algorithm, because here we are using an atomic level of granularity. So the conformer is defined by graph, where the vertices are the atoms of the molecular system. We have seen that when we have a very flexible molecular system, or especially when we do simulation at higher temperature, we'll have too many hydrogen bonds that appear and disappear along the trajectory. And since this is our definition of conformational change, this is considered as a conformational change, but it is not the case because we have just a vibration around the threshold value. So it becomes difficult to quantify what are the relevant conformers. So we have thinking about a new way to represent the conformation. It is a coarse grain representation where now we don't consider the hydrogen, we don't consider the bonds, but we consider the hydrogen bonds by, we replace the hydrogen bond by the cycles or the rings that I form. So since we are working on the peptides, so we have a connected component, we are sure that when we have a hydrogen bond, we will have a ring that will be, at least one ring will be formed. So what we try to do is to, from this 2D graph that we have already on our first algorithm, to identify so the different hydrogen bonds that has been identified during the trajectory, and then try to extract a basis of cycles. And this is one difficulty because there are several manners to get this basis of cycles. We have used the orthon algorithm to get this basis for the time being. And then the idea is once we have the cycles, what we have observed is that some ones play the same role on the molecule. They more or less have the same impact on the 3D structure. So we got the idea to apply what we call the polymorphism. So we try to define the cycles that they play the same role on the structure. And here, what we do is to merge those cycles based on a set of roles. For example, we will merge the cycles that are that have the same interaction with the other cycles, that share at least one atom on that share at least one atom involved in hydrogen bond, etc. And then we get at the end what we call a polymorphic graph. So now the vertex of the conformation will be designed by this graph of polymorphic cycle. So now the vertex is not anymore the atom of the molecular system, but a set of polymorphism cycles. And the trajectory now, it can be seen as a sequence of those graphs of cycles. And here the idea is that from one conformer to another is for the first one we get when we have a change of bound, we will have a change of conformation. Now it will not be the case, because if we change the identity of the cycle, we will have if we change the identity of cycle, we can have the same graph of cycle, the global one. So we will have the same conformer. And we can compare
Sana, sorry, 2 minutes left.
Ah, okay. So
okay.
I will go fast.
Sorry.
So for this, if we compare, here we can compare, and we see that we reduce the number of conformer and transition that have been explored along the trajectory. So for the third application now is the third application now is destined to the vibrational modes. So what we try to do is to from the ER spectrum construct a colored, weighted, and directed graph, where the vertices now represent the motion, the internal coordinate. So we will have distance, angles, and torsion, and we use color to distinguish those types. And the edges will be the correlation between this internal coordinate that we call the cross terms. And we have weighted, so we have weight on the edges. This represents the percentage of participation of each motion on the active bands. So we are interested on the different peaks of the spectra, and we try to get the different motion that have been identified. Here I give you quickly two examples. So we have two monomers, and here we can see that they are participated with the same type of motion. So we have torsion, and they have the main motion that is contributing to the active band is the same for both. And the idea of this is that the graphs show directly the motion that are participating. And we can also, since we are using graph, we can compare between them. So we can compare for a spectrum if we have the same motion at each active band between two molecules, if we get also the same. And also if we go, for example, in the diamer, here we can see that we keep the same, not all the motion that are participating in the monomer, but we keep some of them. And we can see that we have an external, an extra contribution of the hydrogen bond that that we have between the two monomers. So to finish, we see that with the graphs we can do a lot of things. For this time, I cannot show you in details the different applications, but hope you have understood at least some of them. And I would thank the different members of my team, and thank you for your attention.
Thank you very much, Anna. Are there any questions from the public present? Remember, you can ask your questions on the Q&A box.
Hi, I have a question. So
Yeah.
I'd like to know what is the limit of the method? What do you think it could be applied to proteins, or what would be the length, the maximum length of proteins?
Yeah, this is what we want to do. We didn't do it yet, so we don't know if it will be the limit or not. For the time being, the biggest system that we get is the one for the interfaces. So we have a few thousand of atoms, and it works. So I don't know for the protein, but it will be interesting to try on the protein and see what it will give for the both methods.
Can I also ask something? I was just thinking, maybe for proteins it could be simplified instead of considering every atom. you could consider maybe amino acids
Yeah.
as a
That
whole
will
thing.
be a way.
I don't know.
Yeah. So we go to a coarse grain
Coarse grain,
representation
yes.
where we will not be at the atomic, but consider like a part of the molecule as the vertex. Yes, that will be a good idea because this simplifies the 2D graphs and then the different analysis behind this. Yeah.
It sounds it's going to take a lot of computational resources.
But it is interesting to see what you can do with this, because I have seen that for the different presentation before, they were talking about molecular dynamics and they try to get structure. So since this is a post-processing tool, it will be good to see what you can get from this.
Yeah, definitely.